Hi, everyone, and welcome to the Genetics Podcast. I'm really excited to be here today with Dr. Peter Würtz, who is the Scientific Director at Nightingale Health. If you've listened to our podcast with Professor Sir Rory Collins of the UK Biobank, you probably would have heard him mention Nightingale as one of the things he's really excited about for the future. So I was really excited to get the chance to talk to Peter. I've been following Nightingale for a long time since I first heard them mentioned in a talk. I think it was from Mike Inoue at Cambridge, who's a cardiometabolic researcher. And he also thought that the platform was the future of how metabolomics will be measured and make a valuable contribution to precision medicine. So we're going to go into what all of this means. But the company Nightingale also, I think uniquely, has a consumer-facing test that allows people to measure things like cholesterol, triglycerides, branched-chain amino acids, all sorts of things. So they're quite a unique company in that they have an exciting scientific platform and technology, but also have not just research applications, but consumer applications. So Peter, first of all, welcome to the podcast. And I wonder if you could just start by telling us about yourself, your background, and how you found your way to Nightingale.
Yeah, thank you. It's a pleasure to be here. I grew up in Denmark and studied chemistry and physics, and slowly drifted into biotechnology and moved to Finland because of my wife about 15 years ago. And there I got into studies of proteins and biophysics, doing my PhD. One of the best ways to study protein structure and composition is using a technique called nuclear magnetic resonance spectroscopy, which can measure the detailed structure and composition of proteins. So that's kind of how I came to Finland. But then when I was finalizing my PhD, I was intrigued to go into more medical applications of these technologies. And I was lucky to get into a group research team which has just set up a high throughput platform to measure thousands and tens of thousands of blood samples using this nuclear magnetic resonance spectroscopy technique. So I thought that was a kind of a nice way to combine my expertise in measurements of large sample numbers and computational analysis, but facing a more medically applied research area. And it turned out that this technology that my fellow co-workers had set up, it worked way better than anybody had anticipated. It was almost right from the beginning a big success in large population health studies, mainly studying the molecular mechanisms of heart disease and diabetes. But just around the time when we started on the university settings, around 10 years ago, a revolution in genetics was happening, where it was possible to measure for the first time tens of thousands of samples with genetic data. And many of the leading figures in genetics and genomics, they were keen to combine the detailed blood measurements that our technology using nuclear magnetic resonance could do, together with the genetic data. And they had seen from earlier genetic studies that the larger sample size you had, the better the science, the better the confidence in the results, and especially if you could combine the results not only from a single large study, but combining multiple studies from different places. And the Nightingale technology that eventually emerged out of these efforts is exactly doing that. It could measure blood biomarkers and metabolites and lipids at the scale of tens of thousands, and the results were really consistent across different studies. So it was a perfect match to integrate together with genetics. And once that took off, in particular in the Netherlands and the UK and in Scandinavia, many of the leading researchers in genomics, they told their friends that this is great. And then we were overwhelmed with demand, and it was very clear that there was kind of big business opportunity also to set up a company out of this technology.
When you started working with the UK Biobank, for example, what was the initial goal to do the profiling on 10, 20, 30,000 samples, see what comes out of it, and then make a plan from there? Or was it clear from the beginning that this could be applied across the whole 500,000 people?
Yeah, so in the beginning, we were measuring sample sizes of a few thousand, maybe up to 10,000. That was kind of for a long time the biggest studies that were technologically possible to measure. And then the different studies, they combined together that you could get to sample size maybe 20,000 up to 50,000 is still many of the largest studies that have been published so far are around that sample size. But we had the opportunity to work together with the team in Oxford, that many of the people who set up UK Biobank, and we established an on-site magnet in Oxford, and the professors there, they saw that the technology was really good, both in delivering scientifically relevant results, but also in terms of the analytical accuracy and the scalability. So I think that helped everybody to think, okay, what could we do as a next step? Could we one day even measure all 500,000 samples from UK Biobank? And that was for me and many people for a long time, like the scientific goal that we would one day measure all of the samples. And the UK Biobank has a policy that they don't split the cohort into subsets of samples, that they measure case control studies with 5,000 or 10,000 here and there. So when we first had discussions with Sir Rory Collins, he said that, well, come back when you can measure 500,000. And then maybe 1 or 2 years later, we came back and said, okay, look, we believe we are ready if you're ready.
So what kind of information do you get? Obviously, you have the genetic data that you only need to measure once. And then what you all provide is it a small handful of proteins that you're looking for? Is it thousands? Is it hundreds of thousands? And what do you then do with the output data? We could take an example like if you're looking at cardiometabolic disease or something like that.
Yeah. So how the Nightingale blood test works is that it takes from a single sample with essentially no sample preparation. All we do is add a buffer. We measure like a kind of molecular signature of what are the metabolites that are highly abundant in the blood samples. This could be lipids, a few proteins like albumin, which is the most abundant protein, some small molecules like amino acids that are freely circulating in the bloodstream, ketones, and then fatty acid composition like omega-3, omega-6. All of this we get from a single measurement. And then researchers can either analyze the individual data, like say amino acids against cardiometabolic diseases like diabetes, but they can also use the full information of the whole biomarker profile that comes out of Nightingale test to get even better predictive information of, say, future disease risk. Because diseases like diabetes and heart disease, they're not only reflected by a single measure like the lipid levels or glucose levels, but really many molecules in the bloodstream are indicative both for the current health, but also as much for the risk of a progression to more severe disease states.
Well, you had a research paper or a preprint that you put out on medRxiv really recently, which I thought was an amazing exemplar of this, where you profiled, I don't remember exactly how many people it was, but you profiled people who had COVID-19 and were able to predict whether they would be more or less severe. And you also were able to predict, for example, to some extent, who would get severe pneumonia and who would recover relatively safely. How did that come about? It seems like an amazing thing, and I know you're rolling it out in Finland at least to offer this testing to people so that you can better triage who needs urgent care and who doesn't. Is that right?
That's right. It's an exciting study. Traditionally, Nightingale's technology has mainly been used to study diabetes and heart disease, and that was also the consumer product that we rolled out in the beginning of the year. That we're testing heart aids and risk for future diabetes. But there have been some scientific studies earlier showing that some of the biomarkers are also indicative of the risk of, in the future, getting severe infections, being hospitalized for infections, or even dying from infections. That could be pneumonia, it could be also sepsis, and kind of other, some of the most severe complications of getting infectious diseases. So we thought that it might be possible that also the overall blood biomarker profile that we capture for Nightingale could be indicative of those people who, if they get the COVID-19, that they will have a more severe form of disease or be unable to respond in their immune system, and hence get a more severe form of the disease, be hospitalized, or perhaps even die from the disease. Whereas other people who have a more strong immune response, that may be reflected in the overall blood metabolic profile, that those could then be the ones who are less likely to develop more severe disease. And with the UK Biobank, we had for the first time the possibility to test this hypothesis, and the timing worked well by various coincidences that we had the planned release of the first set of data from UK Biobank with a little over 100,000 individuals. That was released in end of May 2020. And about the same time, UK Biobank also released data, the first data on COVID-19 status for participants in UK Biobank. So it happened that around 200 individuals or so in the subset that we had measured with the Nightingale technology, they were hospitalized for COVID-19. And then we were able to show that indeed the overall blood metabolic profile, not a single biomarker, but the combined information of an overall molecular signature or blood biomarker profile could actually predict even kind of who would get eventually hospitalized from COVID-19. And this is despite that the blood samples in UK Biobank, they were measured around 10 years ago. So it's clearly nobody back then had infection when they came to give the blood sample, and of course, certainly not COVID-19. So there is, what we see is that there's clearly something in the overall blood metabolism that reflects if you get a disease like COVID-19, like pneumonia, like sepsis, do you have a response, a defense system to get it? And it works surprisingly well. It's kind of, I think it could be something that could not only be helpful for COVID-19, but we have now all heard that also yearly influenza season is kind of very has a big impact on the health of the every population. And this is actually could be a tool that we can identify the high risk group. So what we see overall is that there's around 10 to 20% of the generally healthy individuals who were enrolled into UK Biobank who had this overall blood biomarker signature that indicates that if they get a disease, it is much more severe than for those who don't have this kind of metabolic signature reflecting the immune response. So we believe this could be a really powerful tool to prioritize who can go back to work or who should better stay at home, who should be very, very careful in terms of their social distancing. And in particular, now that vaccine results already looks fairly promising, we believe that this could be also a tool to prioritize who is seemingly healthy, but would actually strongly benefit from being a first line for the eventual vaccines that are hopefully coming already. So we believe this is a powerful new tool to complement the exciting developments in antibody screening, and these things are kind of rapid trials. Once you have an infection, do kind of how likely are you to get the disease? But we are bringing something new that you can actually, from your molecular profile, identify before you get the disease if you are at risk.
That's incredible. Do you have any insight as to what it is precisely that's driving these major differences? I know you mentioned the immune system, but is it that it's maybe not mounting a response quickly enough, or is it that it's mounting too much of a response? So because I know you can get these cytokine storms that then the body essentially overcorrects and unleashes too much of an immune response. Do you have any understanding of what might be happening?
The exact molecular mechanisms are poorly understood. This is an area of active research. There are some earlier studies looking at some of the key biomarkers that are inflammatory proteins that are kind of like known in the scientific literature, that if you have chronic inflammation, your possibility to have a kind of a strong immune response may be impaired compared to if you don't have chronic inflammation. But the markers of chronic inflammation that are captured in the Nightingale profile are slightly different from the ones that are traditionally measured, such as high sensitivity C-reactive protein, that is a good predictor of future disease, but it's very responsive also to acute infection. And the protein or inflammatory biomarkers that we see from the Nightingale panel, they are shifting very little with acute infection. They are kind of more stable if you do the measurements, say 1 week later or one month later. You generally have the same profile, whereas some of the more reactive proteins, traditional inflammatory protein markers, they are hard to use for preventative screening settings, because you may have a high readout, and then if you come back a few weeks later, you maybe have a low readout. So as a screening tool, this works well, but there's kind of more and more new questions that we open every time we do studies like this in terms of the molecular biology, that I think we are kind of just scratching the surface and understanding what it is. And one fascinating aspect is that it's not only the sort of textbook inflammatory biomarkers that we capture, but it's as much, for instance, the fatty acid balance in the blood, say how much of omega-6 fatty acids compared to monounsaturated and saturated fatty acids in the blood, that also reflects this kind of immune response. It's some of the amino acids that have been linked with diabetes and other diseases, but they don't kind of, the overall signature of infection risk or the immune response doesn't follow the same pattern as, for instance, the risk of diabetes. So many of these biomarkers that we measure, they are definitely not textbook measures of immune system or inflammatory biomarkers, but we have seen both in the UK Biobank and also earlier studies from across Europe that this is kind of something that we see consistently in many different studies.
Do you have any understanding of whether this kind of predisposed state is something that could be modified by changes in diet, lifestyle, taking something, or is it that 10 to 20% of the population, for some intrinsic genetic or otherwise reason, have a immune state that for whatever reason predisposes them to more severe COVID-19 or some of these other diseases we've discussed? Or is it something that people arrive in that state by, based on, the fact that you mentioned omega-6 and omega-3, I wonder if it does have a link in some way to the dietary intake of omega-6 and omega-3? I realize some of this the answer might be we don't know yet, but I'm just curious about how much you do know.
Yeah, it's an intriguing question. Overall, the blood biomarker profile is quite modifiable. Say if you lose weight, if you change your diet, it is possible to change these biomarker levels. So it's certainly not so that you're stuck with a high level and there's nothing you can do about it. In contrast to, for instance, polygenic risk. But since some of this is reflecting inflammation, it's hard to modify with, say, existing medications. There's like nothing like the same way as a lipid lowering medications, where we know exactly we have a pill that lowers LDL cholesterol and we know the mechanisms how it does that. It's kind of commonly the sort of standard advices about living a more healthy lifestyle, lose weight, more healthy diet, that we still give as advice to improve your overall biomarker profile. But this is also an area of a lot of research, and it may be that there are specific also medications, for instance, anti-inflammatory medications that are now coming to market, or even certain dietary supplements that may partly improve this overall biomarker profile. For instance, omega-3 have now been shown in cardiovascular health studies to kind of improve or lower the risk for high dose of certain omega-3 fatty acids. So I think this is kind of nicely complements kind of what you can track yourself and see over time what works for you. It may be not the same lifestyle change, say more physical activity, maybe that works for some dietary changes work for some people. So as we don't have a single solution for everybody now, one can then see kind of over time, and they also get motivation by doing the kind of continuous blood testing to see if you can get yourself more on the right track. So there's two big questions kind of on the people who are mostly healthy, and then they can still improve, and then those who are already, say, have subclinical disease, or kind of maybe older individuals, and the doctor may need to make some decisions. I think there it's maybe not necessarily the same solutions that you would advise as you would for generally healthy individuals.
Yeah, that makes sense. I'd really like to talk more about your consumer proposition and how that works. I had one more COVID-related question, which is there's this emerging concern about people who have many are calling long COVID, which is you have the acute phase, there's recovery, but then for many people there's weeks or months of episodic severe symptoms, shortness of breath, fatigue. Are you all looking into whether there's blood biomarker signatures either before or after infection that can help identify people who are more or less likely to enter that long COVID state?
We have primarily focused on the preventative screening scenario because that's where we believe that we have something really unique to offer. But now that we see these long-term effects, I agree that it would be very interesting to also study these because we see again and again that it's not just a single disease that is affected or a single type of symptoms. So the overall comprehensive metabolic profile that Nightingale captures, I think, can perhaps better reflect this kind of risk than if you're just looking at sort of each symptom individually. And we are in discussion with some investigators in the UK about studying this, but there are no results yet.
That's great. I'm really interested in hearing more about the company decision to go from primarily a research-facing application into something that's more clinical or even now, as of probably about 2 years ago, consumer-facing. I've heard you say before that it's really about closing that loop between discoveries that are made in the UK Biobank, for example, and making sure those get to the clinic and then ultimately to consumers as rapidly as possible. And that's what I really like about your model, because you have this end-to-end discovery all the way through to applications. How do you think about it, and how did that decision come about for Nightingale?
Yeah, so this was the decision we made about 5-6 years ago among the founders, that we really want to take this technology beyond the success in a research setting and trying to make a bigger impact on preventative public health. And we believe this is a technology for primary care, for consumers, whereas many of the other new exciting omics solutions, they maybe first come to specialized care, say sequencing or very, very detailed biomarker profiling with some other tools. We believe this is kind of more like a screening technology, and we want to show that the costs are so low per sample base that this could come out and be used very widely. So that's we made a decision, and we had some of our initial investors. They were keen to see if we could get all the way to that, and that has then been the drive and the motivation also for all the many new recruits that we have had over the years.
So what does it actually cost to do a single sample, and what is the vision for the future? Would you be doing something like this every day, every week, every month? What's the cadence that if you wanted to do a kind of true preventive screening, population scale preventive screening, you could do it?
Yeah, so we have a Nightingale owned clinic that operates in Helsinki, and one can walk in and donate blood, and a few days later you get the results back on your app. And the price at the moment, I believe, is €79 for 2 tests. So we sell this always as a package, because we believe that the tracking yourself is really the key, not as much as what are your levels at the moment, but are you on a track to improve, or are you on a track to get worse? So the kind of continuous tracking, that's a very core concept for this technology. The how often you should measure is maybe not that well studied. We have seen from intervention studies that are, for instance, changing diet, that you see effects already in a 4-week or 8-week interventions. So maybe like every 3 months or something like that would be good. If you do like more like, say, occupational health screening or a national type of screening, then maybe every year, a little bit like a cholesterol test that are many countries have, like a screening cholesterol levels or blood sugar levels widely. This would be natural to complement or even replace those kind of screens.
Yeah, I guess here in the UK and in many parts of the world, there's a kind of equivalent metaphor for MOT testing of a car, where every year you have to take it in and get the yearly checkup. And it seems like you said, on a yearly cadence, having something like this could have profound implications. Where would you see the kind of lowest hanging fruit in terms of impact? Is it in heart disease or cardiometabolic disorders, or somewhere else?
Yeah, so initially we have studied many cohorts and biobanks and trials that have been focused around heart disease and type 2 diabetes, partly because we have a strong lipid profile in the biomarker panel. So it was a kind of a natural place to start in terms of understanding better disease mechanisms. But in particular, in terms of heart disease, there are also existing guidelines that saying if you calculate the risk for developing heart disease over, say, 10 years, and if the risk is over a certain threshold, say 10%, you are indicated for lipid lowering medication. So if you can show that measuring more detailed biomarkers can improve that kind of risk, there's already a model that information could change the decision making for the doctor or the patient. So that has been often like a one of the first examples that have been studied, because other diseases, say autoimmune diseases, it may be that you can identify a risk, but it may be harder to kind of see what can you actually do about all kind of the clinical decision making. As a doctor, you see the risk and discuss with the patients, but will you alter what you do? I think heart disease is probably one of the first areas, but from and we have some evidence, and we're working together with a group in Cambridge to showcase also how many people we can better classify in various risk categories. In a similar manner, that studies have been done with other biomarkers and genetic data. So we definitely believe we can kind of identify some risk groups that would otherwise be missed. There's no doubt about that. But I'm personally now more excited about all the other diseases than heart disease and diabetes, where we see, for instance, the infectious diseases, the new COVID preventative screen is fascinating, pneumonia, sepsis, renal disease, liver disease, and even some of the autoimmune diseases, where there's maybe not the same tradition for doing such large-scale biomarker studies and preventative actions. So we are keen to work together with leading investigators on a scientific level, on a kind of what you can do as a doctor, as a patient, as a consumer, with this kind of information.
What do you think it will take to realize this vision of a kind of truly population scale, yearly or quarterly test? What you said sort of reminds me of similar challenges in the polygenic risk score space, which is there's a lot of evidence that you can identify people that are at high risk for coronary artery disease or heart attack. But it's another thing to convince the national health system or the people in the clinic that you should then, genotype everyone once and integrate this information in clinical care. Are you seeing a similar kind of requirement where you need to now start working with hospitals or healthcare systems to prove that the model works in practice?
Yes, I think there are many parallels to the development in genetics, and we have been following and sometimes copying with pride some of the developments in genetics, where, for instance, in the early days of Nightingale, we were one of the few large-scale biomarker assays that could easily replicate across cohorts. The large sample size kind of made possibilities for kind of new discoveries there of also not so common diseases, and going then to UK Biobank and showcasing with the largest data biobank out there to really showcase that there are these risk groups that we can robustly identify. That's kind of very similar to the discussion in genetics, and likewise the discussions about, okay, this is nice for scientific discoveries, but what is the impact for on a clinical setting? There are also many parallels, and I think the healthcare system in Scandinavia is maybe not the most one that is kind of pioneering such new solutions. So maybe places elsewhere, like in US and Asia, we have many discussions where there's a kind of the payers are looking at this from a different point of view, that it may be not so that this changes the decision making for the individual cardiologist, but if it saves money for the hospital, then it's very clear decision to go for this widely.
That's right. No, it makes complete sense. And is it the same with the consumer proposition? I know you said you're active in Helsinki. Is it an at-home test that people do, or do you have to actually go into a clinic to draw blood? And what stops you from offering it around the world?
Yeah, so at the moment, it's a venous blood sample that is drawn. But what we are now very excited about is the launch of a self-testing kit that we are rolling out in the autumn, and that can capture not all the 250 biomarkers from the Nightingale panel, but crucially the most important ones. For instance, for the COVID-19 preventative screen, we can capture the most important biomarkers, and the same with the most important biomarker for some of the other diseases. So I believe this solves many of the logistic challenges that has been maybe preventing some of the wider scale rollout earlier with easier testing solutions. And we recently opened a lab in US, and also we are now opening Japan. So kind of getting also on site with some of these big markets that may be more amenable to new biomarker solutions. I think that will very much accelerate the entry to healthcare settings.
Amazing. And I imagine that the COVID-19 test you're making could be a real force for bringing this forward into the conversation, because it's such an urgent priority. And if you can prove that it's effective here, like you say, you can maybe do the same with influenza or sepsis or all these other groups of disease. So it seems like it's a good opportunity to not just solve the problem at hand, but also to kind of move the whole field in a more preventive and less reactive direction.
Yeah, exactly. I think part of this is kind of a mindset change that we need. We need to show the good evidence, but that is rapidly accumulating. But many times it's more about people don't even realize that this is possible to measure before people contract the disease, and it could have such a strong information about the future risk. So I have no doubt that this will find widespread applications. But kind of we need to work with the right partners to roll this out widely.
Absolutely. It seems like the sky's the limit. So I'm just conscious of time here. I know you've got a lot of other important work to do today. If people want to keep track of you, Nightingale, the work you're doing, I know they can follow you on Twitter. Is there anything else you'd like to shout out if you all have positions open for people to join your team, or I know you're always looking for great research collaborators?
Yeah, exactly. So we always looking for kind of for talent. Many people in Nightingale, they have a strong background in biobank research, genomics, predictive models, and then the passion to really move beyond the focus about the next publication, but rather the impact of kind of that publication or kind of commercial rollout can really give. So and now that we are looking more and more in the US and Asia, we're keen to kind of also kind of enhance our operations on site there. So you're always welcome to email me if you're interested in these topics, or kind of we have a website that also covers a bit more, many details about our consumer service, and then we're kind of also planning clinics many places around the globe.
Great. Your his Twitter is at Peter Würtz, WYRTZ. I see you've helped those of us who don't know how to pronounce the U with the umlaut by changing it to a Y. Is that right?
Yeah, that's right.
Great. Well, thank you so much. Congratulations to you and the rest of your team, and thanks for the incredible work, especially on the company in general, but the COVID-19 work in general. I'm sure you're working around the clock to make it happen. So hopefully this is a ray of sunshine where we can use this to find our way out of lockdown and into a hopefully slightly brighter future.
Thank you. I appreciate it. Pleasure to be here.